We're very lucky to have Dr. Michael Tomasson here to speak to us today. He did his medical degree and residency at Stanford and then his fellowship at Harvard. Came here in 2000 and is Associate Professor of Medicine and Genetics in the Division of Oncology section of bone marrow transplant and leukemia. He has a very strong interest in multiple myeloma, and he's going to talk to us today about the genetics of multiple myeloma.
Hey, thanks very much. So this is a real honor and a privilege. Thanks, Dr. Herby, for inviting me. To paraphrase a stand-up comedian I once heard, I've suffered for years for my science. Now it's your turn. So here's a rough outline of what I'm going to inflict on you today. So I figured, preparing for this, it's sort of de rigueur to talk a little bit about the history of the field. So I'll just have a few slides about historical overview of multiple myeloma. Sort of the meat of what I'm going to talk about is and you can fall asleep, feel free to fall asleep for the middle bit here. It's about the genetics of particularly what my lab is interested in. And then I'll have a transition with the end, which will have some unpublished data to talk about, just a couple slides, but give an overview about where I think we're trying to take our myeloma program. So this is in the days before HIPAA. This is in the 1850 sometime, the first reported case of multiple myeloma. Sarah Newberry, her name was, again, didn't have to sign HIPAA forms back then. And she was treated to make a real clear distinction between the old days and now. She was treated with rhubarb and orange peel. And you can see her, this is her sternum. The section was filled with this pulpy red mass. She had fractures of all of her long bones at the time she died. And here's a cartoon of just showing the bony erosion of her bones, which is characteristic of multiple myeloma. So this is what I meant by the history. This was really kind of fun to go through and look at some of this stuff. There's only one thing in here that you have to remember that I learned preparing for this talk that I really liked. And that's down. I'll get to that in the bottom here. But this is Henry Bence Jones. So he was a urine chemist, and he was sent some of the urine from one of the first reported cases of multiple myeloma to try to understand what this protein was in the urine. And the interesting thing about doing this history is it's interesting about how history remembers you. So in his, so he has, now we know his, the Bence-Jones protein in the urine has this name. At the time his obituary was written, they didn't bring that up at all. His obituary was written, they talked about it was really important to look at the urine for renal involvement of tuberculosis. So that was what he was known for at the time, but now we remember him from Bence-Jones protein. Another sort of interesting case, so here's a physician that had another case, wasn't the first case report, had a very detailed case report of multiple myeloma autocollar. And so for those of us who are a little bit older, you might remember learning about multiple myeloma as Collar's disease. So for many years it was called Collar's disease. We don't do that anymore. So he's sort of maybe a little bit lost to history. And then Jan Waldenstrom. So Waldenstrom's macroglobulinemia is a different disease than multiple myeloma. It's related. But the interesting thing I think about and what the contribution that Jan had for this field is that he was the first one to refer to the protein in both the blood and the urine of patients with multiple myeloma as monoclonal. And that's a huge milestone in thinking. They knew there was extra protein. They knew there was a so-called church spire, the M spike. They saw that on electrophoresis. But he was the first one to come up with the idea that this was because it was a monoclonal peak. And this is the thing, so it's just one thing to remember from all this, just because I was so tickled by it, was this Ukerlati test, I think it's pronounced. It's a Swedish physician who came up with this passive diffusion test. You put in an antibody in the center well here, and you put different antigens of different dilutions around the outside, and depending on those arcs of precipitation, you can characterize the antibody was there. So a scientist, Dr. Korngold, and his technician, Dr. Lipari, did this with the Bence-Jones proteins and found out there were two general classes of Bence-Jones proteins. They weren't all the same. They fell into two groups. And because of that observation, we have the name of kappa and lambda named after Korngold and Lipari. So that's a neat little historical fact. So I'm not going to dwell on this too much, and then now skip ahead, 100 years worth of immunology. I won't go into too much detail, and that looks a little unfocused to me, but maybe it's on here. Yeah, I think this slide is okay, but so we now know that a lot about the structure of monoclonal proteins and of antibodies. But I'll just say one of the intriguing things and why I like the fact that Jan is up here is, it doesn't, there's many different types of antibodies that are secreted in multiple myeloma, and there can be fragments and there can be light chains and heavy chains, but it's the monoclonality of it that ties it all together. So I think that, for me anyway, one of the confusing things is that you don't have one particular antibody expressed, but just each patient has a different one, I guess is what I'm trying to say. So again, just to give a broad overview of what we now call plasma cell dyscrasias, and again, I apologize if this is all very clear to you, but it wasn't clear to me when I was a house officer, that this is really a spectrum of diseases that are all arising from this abnormal monoclonal plasma cell. So this is, for those of you who don't read the stuff, MGUS, monoclonal gammopathy of uncertain significance. How could you not study something with such a name? Is a sort of benign precursor lesion, and we're going to come back to that at the end of the talk. And so there's something wrong here. There's a clonal development of these plasma cells, and depending exactly on how things go from this stage, you can have different flavors of disease. So systemic amyloidosis and light chain deposition disease are essentially MGUS cases that have a toxic protein secreted, and they could be a light chain or it could be an amyloidogenic protein. And the disease is really coming about from the secretion of the proteins, not so much of the behavior of the cell itself. Whereas you have the malignant form of multiple myeloma that has the classic CRAB, the hypercalcemia, renal involvement, anemia, and bone destruction. So here's really, for those of you, for the cardiologists in the audience, just to a review of multiple myeloma. Here's a fracture. This is a patient maybe who didn't to break their arm skiing, but to just reach down to grab a cup of coffee and broke their arm. You've got sheets of plasma cells sometimes in the bone marrow. These should be very widely spaced in the bone marrow. And here's a classic plasma cell, and you can see the chromatin is very clumped. This was described very early in the course of the disease. And here's SPEP, for those of you who haven't seen it. Here's a monoclonal protein compared to normal control. This is just serum of a patient run out on a thin agarose gel. And this is a little bit about the epidemiology, and one point to take from this that I'll, again, return to later and I think is interesting for the genetics, is that we know this is African Americans over here, and here's some other ethnic groups over the age. Number one, there's no kids with multiple myeloma. It's a strictly disease of aging. And number two, we know African Americans, we don't know why, but they're at about twice the risk of developing multiple myeloma as whites and Asians. Okay, another take-home point that I'm not going to dwell into, but just for those of you who don't want to know something about multiple myeloma, especially, again, for the people who are been around for a while, multiple myeloma was sort of this orphan disease in a way, because for decades, this is a Kaplan-Meier showing our progress over the decades. For decades and decades, this was a dismal disease with not a lot of hope. And you can kind of see here, in the 90s, we started auto-transplanting people, and it's kind of remarkable how little effect that had on survival, but a significant effect on survival, which is why we do it. But then with the advent of bortezomib now, Velcade, it's a huge revolution, and this is helping our patients live longer. So the survival curve for this disease has been bent by a lot of work, and I'm going to dive into the science here for a minute. All of this progress has been made empirically by physicians and clinical researchers, so it's always very humbling for me to try to come up with something from the lab that can contribute to this progress. Okay, so why study? This is not multiple myeloma. These are giants in the field of chronic myelogenous leukemia, but just as a general heading, I study chromosomal translocations, a particular chromosomal translocation in multiple myeloma. Why do that? Well, this is just one slide just to show Janet Rowley and Brian Drucker. Janet found that the Philadelphia chromosome was actually a chromosomal translocation that creates the BCR-ABL in CML. Fast forward 30, 40 years, Brian put the first phase 1 study of imatinib into patients. And it's interesting from a couple of perspectives. One is that the phase 1 trial was a failure because, as you know, the design of phase 1 trials define the dose-limiting toxicity of a drug. They didn't find it. They ramped up the dosage and never found a dose-limiting toxicity, but all the patients at a certain point went into remission. So he's changed the face of things. The other thing I like bringing up, of course, he's an alumnus of Wash U, And the other great thing that I like the history and the lesson from Brian's story is that he was told when he left Harvard, they didn't offer him a position there after fellowship, and he was told he had no future in science and by the muckety-mucks at Harvard. And so he's on the short list now for a Nobel Prize for his work at imatinib. So why study chromosomal translocations? Because it teaches us a lot about the science, and eventually it may lead to a drug. So for many years in multiple myeloma, we didn't think there were a lot of interesting chromosomal translocations, because if you do the same thing we do in AML or CML, take the cells out and put them in a dish and do routine cytogenetics, not much happens. So we thought, oh, well, there's not much there. But then a few years ago, now 20 years ago or so, Michael Keel and Leif Bergsegel in his lab at the time did southern blots, molecular technology, to look at chromosomal translocations, and found actually they're very common, and there are recurring partners that happen, just like in other leukemias. So this was sort of a milestone in the field. And I'm going to focus, there's a bunch of recurring partners that happen. I'm going to focus just on one, this MM/FGFR3, and I'll tell you why. So this is an old slide. With modern therapies, patients do a little bit better with the 414 now, but it's still basically a bad prognostic sign. This was before Velcade. The patients did a lot worse if you didn't have the 414 versus whether you did. So it's a bad, of the different chromosomal translocations, it's a bad prognostic sign. And here's a nice clinical case. I like to frame things around this. This is a particularly bad case of multiple myeloma, but it's a real one, and it makes the point. 64-year-old man was turned down as a blood donor, felt okay, turned down as a blood donor because of anemia, broke his hip, a pathological hip fracture, maybe he was stepping off a curb, got diagnosed with multiple myeloma, 56% of cells in his bone marrow, plasma cells, and he has this 414 chromosomal translocation. Again, not by cytogenetics now, but by FISH, which is the best test that we do for this. And he progressed through all modern treatments, including Velcade, and died shortly thereafter. So there's another reason to study this, is why do these patients, some patients do very badly. Okay, so moving into what I'm studying. So the chromosomal translocations can be symmetrical in the sense that they're are balanced in the sense that you have two partners going on. You have a MM set, this one partner gene at the breakpoint, versus the other side. And I'm going to not focus on FGFR3, and I'll show you why. The attention really turned to this other gene. And I apologize, it's named MM set for multiple myeloma set domain containing protein. And it's also, every now and some slides, you'll see WHSC1 is another name of this gene. And this is just the structure of the locus, showing FGFR3 over here, and then different breakpoints that occur. And these are the exons of this MM set gene. So don't panic. So I understand that a microarray pictures are like pictures of kids, right? They're great if they're yours. If they're somebody else's kids, then it's not very interesting. But the point here is ignore the p-value over here for a second. And the point I want to make is just this, the FGFR3 one here, is if you look, and this is patients up and down here, and the gold ones are 414 patients, and these are different genes here. You see this MM set gene is red in all the gold cases. You know, it matches up very well. All of them express MM set or WHSC1. FGFR3 is very highly expressed in a subset of these, but there's a group here and a group down here which have lost FGFR3. So that's one of the reasons FGFR3 is a great candidate. It's actually the reason I started looking at this was because it was a good candidate oncogene. But this is the reason why I've sort of moved over to studying MMSET. Okay, and what is MMSET? Well, I'm not going to burden you too much about this because it's a lot of negative data. There's 3 protein isoforms. We thought in the lab, we're very excited because the techniques we use to study proteins in leukemia are very good. So this is Western blots showing the proteins, using an antibody that shows the full length and another antibody directed towards the middle here. So here you only see the long and the short version of this protein. And here, though, if you use an antibody down here, you can detect different breakpoints that give rise to different size proteins in patients and cell lines. So this was very confusing, so we figured we're going to do all kinds of experiments to figure out which one of these proteins is really the driver, which is causing the disease. So here's about an embarrassingly long number of years of work with all these MMSET isoforms. And the kicker for me was expressing this protein in mice that already were prone to getting cancer, and they lived long and happy lives. I'm not going to burden you with this either. This is some of the background of the protein. What the MMSET protein we think really does has to do with DNA damage. It's a bona fide important gene and protein in its own right. But whether or not this has anything to do with cancer remains to be seen. So what did we do? Well, we went back to the drawing board and made a special kind of microarray. And this is what we call a tiling microarray. It's a physical thing. It's like painting a region of the chromosome and saying not looking at particular genes, but just this physical region. Because we know where these breakpoints happen in the chromosomal translocations. So we wanted to say, let's paint this whole region here with probes and then put RNA from patients on there and see sort of what lit up, just to be very unbiased, as we say. So here's the raw data from one of these things, and reassuringly, the thing that kept coming up, this is very noisy because of this type of array, but if you do repeat this on a number of patients, you do see a theme statistically, and it's all this MMSET or WHSC1. This is the consistent region that is dysregulated in all of these patients. But there was another little bit of probes here that was interesting to us. So this is a blow-up of this tiling array, and this is a physical thing that's painting across this chromosome near the breakpoint. And so when you put the RNA from a patient without the translocation or with the translocation, you would expect, gratifyingly with the red, you're getting RNA lit up. It's activated. But again, what these peaks should be the exons, right? And that's pretty much what you see, is the exons of this gene lighting up. And of course, the interesting thing to us was this huge peak over here, which is in the middle of an intron, which shouldn't be making any RNA. So we use these arrays over and over again in patients with multiple myeloma and in cell lines to really prove to ourselves that this thing was real, that it was on, and indeed we think it was. And here's what we think it is. It's a class HACA. Sorry, it's a very esoteric flavor of RNAs. I think I see Gene Schaefer. He's probably the only person that knows about these things in very detail. It's certainly new to us. We did northern blots. This is a real test, again, to confirm, is this really on? Is it really expressed in patients and in cell lines? And the answer is yes, it's on in cell lines. And yes, it's expressed in patient samples and highly correlated to the chromosomal translocation. So just to be clear what we're talking about, on the one hand, you can fall asleep for the science part, but what we think is going on here is a chromosomal translocation involving this chromosome 4. It hits this gene, and we do think MMSET or WHSC1 is the target of this chromosomal translocation. But buried within this gene that everyone else has been studying, between these two exons is this little bitty non-coding RNA that is also at the same time overexpressed. And we were very struck by that, given all the negative data that we had. The other thing that, again, it's tough to get across, but I just thought is too good to ignore, is the fact that these two exons here, this is a very big protein, but it's an enzyme. It's a histone methyltransferase, and that histone methyltransferase is encoded by two exons, this one and this one. And so this RNA is right in the middle of the heart, the beating heart of this gene. So it just seemed too good to pass up. So this is the sort of thing we do to try to prove our bona fides in the laboratory. Is this what type of RNA is it? It's a non-coding RNA. It doesn't make a protein. It's localized in, these are purified nucleoli. So these are a northern blot showing it's predominantly located in nucleoli. And I'm not going to walk through this too much, except to say that coilin is a Cajal body marker. It's another little part in the nucleus to see where this thing lies. We don't think too much of it is located there, but the majority of it seems to co-localize with nucleophosmin, which is a nucleolar marker. So we think it's a nucleolar localized RNA. And for the esoterica here, so SCA is Cajal body localized RNA. It's called that in the databases. We think it's a SNO RNA. It's a broader class of non-coding RNAs. Okay, so I'm almost at the end of the annoying biochemistry, but this is the thing that maybe you need to know in terms of basic chemistry is what do these things do? Well, in generally, what they do is they bind to other proteins, and they guide these protein complexes to a particular target. So that's what presumably this RNA, the ACA11 RNA, is doing, is it's guiding some protein complex to a particular target. And this HACA are very well-studied form of small non-coding RNAs, they bind to discarin complex, and they modify RNAs in your ribosome. So what that had to do with cancer, we had no idea. So we figured, okay, well, we really were sort of stuck at this point, but let's make sure that this is true and see what's going on. So this is a complicated plot here, but basically what we did was we went fishing. We took the RNA and tagged it and put it in some cells and pulled it out and saw what proteins we came out. And originally I told my postdoc, I said, well, if you don't see this discarin complex protein, then the experiment didn't work. And I didn't think the experiment was going to work anyway, so we didn't see that, so I sort of said, I told you so. But all the proteins we did find did kind of fall together in a theme where they were known to bind together through a number of different papers, and they seem to form this theme about an alternative splicing function. I'm not going to talk too much more about that because we don't know too much more about it. But this is my chance to show this slide, which I like. So this is this very trendy thing in biology and biochemistry now, systems biology. So you show one of these network slides and say, okay, thanks to systems biology, we now have a very clear comprehension of complex diseases. Okay, so back to this slide. So what what is this RNA? What can it be doing? Well, one of the clues we had here is this is microarray, and again, I'm not going to inflict too much on you except to say this is patient samples. And one of the things that jumps out, here's the MM set over here that jumps out. This is sort of, we couldn't make rhyme or reason of all these upregulated genes, but the signature here is hugely strong. All these genes, a vast majority of these downregulated genes in 414 patients are ribosomal protein genes. And so here's a nod to our fellow WashU player here. And this is the translation, the original French of the chance favors the prepared mind from Pasteur. And so Dr. Schaefer and colleagues described another SNO RNA that was embedded inside a ribosomal protein gene that had to do with regulating metabolic stress. And so we thought this coincidence was just too huge to ignore. And so we read this paper very carefully and decided to look at metabolic stress in our assays. And lo and behold, ACA11, and again, just for Gene and everybody else, the idea here is that these things are causing, they're the mediators of oxidative stress. They're causing stress in cells to die. But ours is sort of the other way, but our signature was that these might be downregulated. So it sort of made sense to us that if ours is up and ours going up causes these mediators to go down, that might make sense to protect the cells. And indeed, that's what's happening. When you put AC11 up, it causes stress to go down. And this allows the cells to grow more happily. And this is the key thing here, that I think, of what this chromosomal translocation is doing, is it causes resistance to the traditional chemotherapy by protecting the cells from oxidative stress. Okay, and this is just to say that it works the other way around. If you knock down this SNO RNA, AC11, you induce stress, and you make the cells more, they grow less well, and you make the cells more sensitive to chemotherapy. So this is the idea of maybe there's some therapeutic angle here. If we can reduce this, we'd make the cells more sensitive to chemotherapy. Okay, and this is just, I'm just going to point out really, I would, just for Gene, we look very carefully to see in non-myeloma cells, we do see some, is AC11 actually causing downregulation of these other ribosomal protein genes? Some of them, yes, to varying degrees. Interestingly, at the RNA level and the protein level, it does cause them to go down. And here's one that we had our eye on because it was this Gene's paper. It causes the downregulation of RPL13A. And you can see that here. And the SNO RNA within it. So this is our model of what we think AC11 is doing, is really through modulating some of these other SNO RNAs. And I'm not going to talk about that, but we think, I'll just say for Gene really, so here's a pull down of the RNA from this complex, and it's very interesting. Here's a representative SNO RNA embedded inside ribosomal protein gene. And you notice it's interesting. It starts very sharply here at this edge, and then doesn't end sharply. So this is the signature we think of an intermediate. So maybe this complex has to do with processing, splicing out these other SNO RNAs from these genes. Okay, so I'm not going to go through this except to say this is from a companion piece that says maybe what's going on is this acting as a sponge to suck up these factors that are important in biology. And here's a bunch of different biology, including metabolic stress. And by overexpression of our SNO RNA, it's shifting the balance over there. Okay, and so that's the conclusion from this part of the talk. We think ACA11 it's a novel SNO RNA. We don't know its target. It's localized to nucleoli, and we think it helps modify reactive oxygen species. Okay, now I'm going to switch to the second part, so hopefully wake you up. So that's the worst of it is over. So now a little bit of just sort of picture. I'm going to show some unpublished data on a completely different topic, but I don't know if anybody knows this Numbers show. It's on Netflix, and it's kind of a corny show where they have two brothers. One is this hard-boiled cop for the FBI, and the other's a mathematician. And lo and behold, every crime, every murder that happens, there's a need for math in there somewhere. So he gets his brother to help him with the math. So, and so one of these things, so it's kind of a corny show. I kind of liked it. But the mathematician brother one time said something out of the blue that just sort of stuck with me. And that is, you're like a doctor trying to find better ways to treat patients that have been thrown out of car windows. The breakthrough, is not in better medical care. It's seatbelts. And I get a little tired of treating the patients as they've been thrown through the windshield. So the thing that I want to bring up now that, again, is an important take-home point, if you don't care about any of these other things, is that I want to study MGUS. I want to study early in the disease, get past before the windshield here. And why study MGUS? Well, a couple of things have happened that are new and that have prompted us to look at earlier in disease. One of them is we now know that virtually all patients with myeloma have this MGUS precursor. Now, MGUS is very common. Not everyone with MGUS, a very small number of people with MGUS get multiple myeloma. But for years, people would ask me, and one of the reasons I like leukemia and multiple myeloma is people would ask, was there any way we could have caught this earlier? And for years, we say, no, this just comes out of the blue like a lightning bolt. But now we know with multiple myeloma and more of these hematologic malignancies, the answer is actually yes, we kind of had a clue. But we just don't know what to do. The second thing is, I mentioned that African Americans are at twice the likelihood of getting multiple myeloma. There was a follow-up study that looked and said, well, is that risk because they progress from multiple myeloma more rapidly, or do they get more MGUS? And the answer is they get more MGUS. So the African American predisposition to multiple myeloma is actually a predisposition to the precursor of multiple myeloma. And finally, there's this idea that it's really a benign thing. If somebody has MGUS, what do? Well, don't do anything. They're going to die probably of something else because it's so common and myeloma is so rare. But the other breakthrough is now we really know how to identify patients who are completely fine. And you guys, medical people, should know this. So if they're IgG subtype as opposed to IgA or IgD or IgM, if they're IgG and they have a normal serum-free light chain assay, their chance of progressing to multiple myeloma is very low. Whereas if they're non-IgG or if they have an abnormal serum-free light chain assay, they're at much higher risk for progressing. So now we know there's this 1% number, but now we know who are the really low-risk people and who are the higher-risk people. So this is the radical concept that I just want to plant in your mind. So nobody believes this but me, so this is an advertisement, right? So the entire field is focusing on, well, these patients always relapse. What do we do when they relapse? Fine, I don't know if I have a role in that arena. But looking earlier, there's a radical concept. Why study the early stages? Well, is it conceivable that we could prevent multiple myeloma? And this is basically just to highlight some of the people here, our program that we have. We're putting together, there's a bunch of people over the years that I've convinced to get interested in multiple myeloma. So we're trying to come up with a whole team of people to study multiple myeloma, and I'm trying to put this spin of looking at the earlier stages of multiple myeloma on there. And so the last thing about science, my contribution to the science of that prevention, the early stage, is this one. This is actually a mouse SPEP. So mice get MGUS. So this is normal mice here with their SPEP and albumin and different other proteins and the immunoglobulins up here. And here's a mouse with an M spike. And it's a particular kind of mouse. There's a strain of mouse that's been known for 40 years to get MGUS, this Cal Ridge mouse. This is just sort of a map of different strains that we studied. We did all kinds of tests on these different strains to try to figure out what made them different. And we were flummoxed. We knew we had a mouse that had an increased risk of MGUS, but we didn't know how to figure out what the gene was. And here's an example of what these mice do. This is now looking for that M spike, sort of doing screening for MGUS in our mice. Here's the Cal Ridge mice. We take a baseline serum, and hardly anybody has a spike. There's this one
weirdo over here that does, but nobody has a spike at baseline. We immunize them, we boost them, and then we follow their antibodies with time. And just to make this point, I like to look at this AJ strain. The AJ strain has a super response to immunization. It's very hyper-responsive to immunization, but it's after a while is gone. Comes up and crashes. Whereas the phenotype we're interested in MGUS is in cow-ridge mice, it doesn't have too big a response right away, but it never goes away. And that's different from the parent black 6 strain. So we had sort of a breakthrough when Sarah Amund in Kathy's lab actually started hearing about us complain about how to figure this out. And she knew that we had some data from patients with myeloma and without myeloma, and we're trying to figure out what the genes are at risk for multiple myeloma in the patients. And she knew we had this data from the mice. And she had this idea of trying to compare these two data sets and sort of do a Venn diagram. And the first critical thing that she did, that she was able to do, was to go through and compare strips of DNA haplotypes that are different from the cow-ridge and the black 6. And if you do that, you can come up with sort of a map of these clumps of DNA that are different between these two strains, and we think the answer is in there somewhere. But it's still a huge bin to look in. And this is now the saying that I never understood, but now I do, is it's better to be lucky than smart. This comes in here. She looked at the human data that we had from a little bit more samples than this, but from patients with and without multiple myeloma. She compared these. She had a bunch of genes that overlap, still way too many, and I thought we were going to publish the paper there and just say, well, 400 genes, maybe let somebody else do the hard work and figure out which one of those genes is really of interest. It's going to be hard to figure that out. But then if you add in a published array data comparing black 6 and cow-ridge mice, there's 71 genes on their list, and if you add that into to the mix here, you come up with just 4 genes, and that's tractable. We can look at 4 genes. And so we really didn't know how to look at the 4 genes, so Sarah started just doing PCR, amplifying these genes. We figured we'd sequence them and try to figure out some little difference. And we came across this Samson, or she came across the Samson1 gene, and it's just gone. It wasn't subtle. Here is just the DNA of the gene, different parts of the gene in the black 6 mice, and it's just missing in cow-ridge. And here's the same data from the SNP arrays. It's just a block that's just gone. And this is what I mean by lucky, very lucky to smart. What is this Samson1 gene? Well, it turns out it's a negative regulator of B-cell activation, which is sort of exactly what you'd think you'd be looking for if you have too many hyperactive B cells in MGUS. And it's been studied by people before, and I'll just say it's a signaling protein. It does make the B cells a little bit hyperactive and gives you more, at least in our mice, gives you more IgG produced over time. And the lucky and ironic thing is that the whole thing was originally cloned in people studying multiple myeloma because it was differentially expressed in patient samples with multiple myeloma. So Sarah's a really good graduate student. She says, we don't know that this is absolutely the thing, but it's a great place to start. Okay, so this is our gang. So I'm leaving a lot of people out here, but there's Liang Chu, which did the lion's share of the work. She's now back in Shanghai, did the lion's share of the ACA11 work. We couldn't do any of this without Ravi providing us patient samples. Dr. Vidge, here's Sarah Amund, who worked on from Kathy's lab, who is actively working on the cow-ridge project. And Len Maggie, who's a very productive collaboration from cell biology. And this guy is interviewing here for MSTP. We're all going to be working for him someday. He did a lot of the bioinformatics just that I didn't understand, and asked him if he could do it, and he did it before he went off to Yale, and I'm very happy, and hopefully he'll come back here to be an MSTP student. All right, I think that's all I have. Thank you very much. I don't know how I did on time. I didn't see. Yeah?
It was
Went
great,
fast.
Mike. Thank you very much. We'll open it up for questions. Dr. Karsh?
Mm-hmm.
Yeah, I think you make a really important point, actually. And one of the reasons you may wonder, why did you make that turn? I have these two parts of the talk. One where I'm talking about is translocation. There's plenty of work to do there. And I made that left turn. One of the dirty secrets of genomics and genetics of multiple myeloma is if you look at the MGUS, you really want to know, MGUS people live with and die with. It's relatively benign. But the multiple myeloma kills you. If you look at genetics and what the mutations are, they're virtually the same in those two settings. So what is actually killing people, we don't, it's kind of mysterious. And one of the theories that I've been trying to lasso my wife into looking at is that it's actually, the answer is in the stroma. That the difference between MGUS, the pre-malignant and malignant stage, may not be a genetic. We like to think in cancer, oh, there's P53 or there's RAS or there's something happening that's causing that transition. Personally, I'm beginning to wonder, because of the issues that you raised, if it isn't really the key step between pre-malignant and malignant, maybe a stromal inflammatory picture as you're describing. Yeah, Alex. Yeah, so I wasn't going to say this, but given that what we're doing, I'm going to throw this out there, political thing. So definitely we have a lot of drugs that need to be tried for patients, and I think I could say, I could make something up, but I'm wondering if we're not coming to the end of the pharmaceutical era of medicine. And so if it does turn out to be a stromal problem and multifactorial inflammatory thing that's going on, there may be other things that we can do, diet and exercise, maybe something else. There may be some non-traditional therapies that we can do to try to slow down this transition, this inflammation that may be causing the root of multiple myeloma. So it's kind of an escape route, but I had to get that out there. So maybe it's something to do, with reducing inflammation. Aspirin, we now know, is a huge makes a big impact on patients with cancer, sort of retrospective data. That's one of the questions we're looking at. Graham Coldis here has some data that aspirin, patients who take aspirin, or not patients, people with aspirin, take aspirin tend less to develop multiple myeloma and multiple other cancers. So maybe it's as simple as that.
Jean?
Yeah, so it's, and again, I'm going to try to stay on this theme because it holds together. It's very specific in the, in terms of the cells. It happens in about 15% of patients with multiple myeloma. In terms of the cells, we know that it's in the plasma cells. It also is found in memory B cells. So there's this germinal center reaction, and we know it's not, happens not earlier than that, right? It's not in the bone marrow. It's not in the bone marrow earlier B cells. It happens in post-germinal center cells. But in the germinal center, you spit off into plasma cells and memory cells. And you can see, and there was Bill Matsui, who was here last week, was thinking about maybe these are stem cells. They're not plasma cells, but they may be feeding into this. So you can see the translocation in both of those cells. But one of them is malignant, and one of them is kind of not. But maybe these other cells give rise to the phenotypic, the bulk population of cells that we see. And what was other? Right. Yeah, not us, but others. And this is another dirty secret, and it gets back to this question of the stroma. The dirty secret is it's pretty much just about everything, including the 414, is also found in MGUS patients at just about the same frequency. So the reason why I think, so you can look at that as a glass of half full, half empty. A lot of clinicians I talk to, the hematologists love to come up to me and say, oh, I have a patient with 414, and he's fine. And so I also have people on study section who say, well, why don't you show us that this causes multiple myeloma? You haven't done that yet. If you look at the patients, it's not causing multiple myeloma. It may be what this is it modifies the phenotype once you get multiple myeloma, right? So
if you get multiple myeloma, you don't want to have one with this translocation because it makes you resistant to your oxidative stress, makes you resistant, and bortezomib doesn't work through that, so they respond okay to bortezomib, but it takes away your alkylating agents, which may work through oxidative stress. And that's a key linchpin in our treatment strategy, and if you don't respond to that, and so that's getting back to Alex's point, maybe, I'm not sure about the treatment part, but in terms of this 414 part, maybe what we need to do, and we already have data that patients with 414 don't do well with autotransplantation. We still transplant them because we don't know what else to do. They don't respond to transplant. We put them this whole procedure. Maybe we should look and say, okay, we're not going to waste our time giving you alkylator therapy because it's not going to work for you.
And Mike, the increased risk in African Americans, is it completely genetic, or do you think there are environmental factors as well that either trigger the increased risk?
Yeah, it's a great question, and I think it's generally, as I understand epidemiology, we think it's genetic, but it's a hard thing to tease out. We also know the other piece of evidence that may push us towards thinking of a genetic reason is we know there's also a slight increased risk of MGUS, multiple myeloma, in families, in first-degree relatives. But it may be environmental, but the thinking is it's genetic. And the other reason for that, the other critical piece is we know also that there's an increased risk of multiple myeloma and MGUS in Africans.
Thanks. Kathy?
Yeah. Well, certainly bisphosphonates, as you know, are standard of care in the treatment of patients with multiple myeloma, to strengthen to stun the osteoclasts and to prevent progression. I think this is going to be tricky, and again, it's interesting to look at the progression because we know that the progression of multiple myeloma gets to a stage where it's independent of the stroma. So, and this is a problem in terms of our drug trials because we want to try, if you want to test some new drug, a stroma drug, you find patients who are multiply relapsed and start there. But these are the patients that are going to be mostly independent of stroma. So I think the time to look at the stroma is as early as possible in the course of disease because that's where it starts, stromally dependent, before it migrates to its independence. So I think you could, but you'd have to have a very forgiving view of the clinical trials because I would say that most of the patients who have relapsed and refractory disease are going to be in a stromal independent stage. Yeah. Yeah, there's one study from Gareth Morgan and his colleagues in Britain that did a GWAS. It was published in Nature Genetics and found some strain, like most GWAS, they find some loci and they don't know what they mean that seem to be tracking. We're doing another, I'm personally involved in a separate African American GWAS as well as another sort of parallel Caucasian GWAS experiment. And so my role in that is we're providing, really Ravi and our team here is providing the samples. I'm going to go with the hypothesis and say, let's look at the SAMPSON1 pathway in the mouse and look, as you know, GWAS is unbiased, and you have to have these astronomical p-values because of that. But I'm going to say, okay, once we get all this data together, let's just look at this one pathway and see if there's any abnormality. And that's a long shot to say there's conservation between the mice and the humans, but it's a hypothesis. Yeah. Yeah. Mm-hmm. Yeah, so it's really a spectrum, right? So what you're referring to, the 10% in the bone marrow, and again for residents, 10% of the bone marrow and 3 grams, these are the major
diagnostic criteria for multiple myeloma. Really, the final ingredient of that is you have to have end organ damage, right? So for the diagnosis of multiple myeloma, you need your bones dissolve, lesions in your bones, or you need renal failure or anemia as end organ damage. So you can have just the 10%, you can have just the large amount of protein without end organ damage, and we call that smoldering myeloma. And so the difference between smoldering and stage 1 myeloma and advanced, there's no question in my mind it's a spectrum. And what differentiates those stages, we don't really know. And I guess what I'm saying, I don't know the percentage in terms of the progression, but it's very high. If you have someone, as opposed to MGUS, we know the MGUSs that don't progress, but the MGUSs that have these features, or if you have 10% or 3 grams of protein, they're at very high risk, meaning 30, 40%-ish. Don't quote me on this, but about that range over the course of a couple of years. They're very high risk of progression. Yeah. Right. No, it's a great. I love love these things. In the history of physicians, scientists, that's the sort of clue that leads to some breakthrough. But no, have no idea. But it's a good point. The anemia is out of proportion to the marrow involvement. We don't know why.
Mike, thank you very much.
Yeah, thank you.